Life runs on chemistry, but it runs at a speed and precision that ordinary chemical reactions could never achieve on their own. If cells relied only on random molecular collisions, life would be too slow to exist. The reason biology works at all is because nature built molecular machines, enzymes and proteins, that control chemistry with extraordinary efficiency. This is the Learning Studio, where chemistry is explained as the operating system of life rather than a collection of facts. And after understanding how biochemistry explains living systems, the next step is to understand the molecules that make those systems work at usable speed and accuracy. Most people think proteins are just structural components or nutrients, but in reality, they are functional machines. Muscles contract because of proteins. Oxygen moves through your blood because of proteins. Signals travel through your nervous system because of proteins. Enzymes are a special class of proteins that act as catalysts, accelerating chemical reactions without being consumed in the process. At the molecular level, enzymes work by controlling energy and geometry. Chemical reactions normally require significant activation energy to begin, but enzymes lower this barrier by binding reactants in precise orientations and stabilizing transition states. Instead of forcing reactions to happen, enzymes make the correct pathway easy, allowing reactions to occur millions of times faster than they would on their own. This molecular precision was first clearly described by Emil Fischer, who proposed the lock and key model to explain why enzymes are highly selective. Later, Daniel E. Koshland refined this idea by showing that enzymes change shape during reactions, actively adapting to their substrates. These insights revealed enzymes not as rigid tools, but as dynamic machines. Proteins themselves gain their power from structure. A protein's function is determined by how its chain of amino acids folds into a three-dimensional shape. Weak chemical forces, hydrogen bonds, ionic interactions, and van der Waals forces hold these shapes together. Small changes in structure can completely change function, which is why mutations can have dramatic biological effects. Enzymes and proteins make life chemically possible. Reactions that would take years occur in milliseconds. Metabolic pathways are tightly regulated. Errors are minimized. Energy is conserved. This level of control allows cells to survive, adapt, and evolve in environments that would otherwise destroy fragile chemical systems. Understanding enzymes transformed medicine and biotechnology. Drugs can be designed to block or enhance specific enzymes. Industrial enzymes are used to produce food, biofuels, detergents, and pharmaceuticals efficiently and sustainably. Even modern genetic engineering relies on enzyme-driven reactions to cut, copy, and assemble DNA. Enzymes also highlight a deeper truth about chemistry and life. Complexity does not violate chemical laws. It emerges from mastering them. Life doesn't escape chemistry, it optimizes it. And once we understand how enzymes and proteins control chemistry so efficiently, the next question naturally follows. If we can design molecules to influence enzymes, then how does chemistry intentionally design drugs that interact with biological systems? That question takes us directly into the next episode, where we explore medicinal chemistry and how drugs are designed at the molecular level.